the life of a leukocyte. And you probably want to.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes
This is
roll
how
slowly
complex
on
things are.
endothelial cells.
This is not simple rules.
P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells.
That means the inside of your blood
interactions are
vessels.
broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell
They
types.
all have little hair sticking out.
The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid
Yeah.
raised above the rest
Tell
of
us
the
something we
lipid
didn't
recruits specific membrane proteins.
Yeah, I saw that on the good
Rats'
just
rigidity
last
is
night.
caused by the tight packing of
I did
cholesterol
have
molecules.
It was on Wheel of Fortune. wasn't it?
So all of this happened by chance and random events.
Yeah, that's Thursday.
Keeps in unsaturated hydrocarbon chains and lower cholesterol.
This is how
concentration result in
white blood
increased
cells
fluid
gets to a wound.
at sites of
Okay.
inflammation.
Wow.
Secreted
Sites of
chemokines
inflammation.
bound to heparan sulfate proteoglycan on endothelial cells are
Here's the
presented to
trigger
leukocyte
to
7
call the
transmembrane receptor.
white blood cell.
The binding stimulates leukocytes and
I cut my
triggers
finger.
an intracellular cascade of signaling reactions.
Chemokines.
The inner leaflet of the bilayer has a very different composition than that of the outer leaflet. While some proteins traverse the membrane, others are either anchored into the inner leaflet by covalently attached fatty acid chains or are recruited through non-covalent interactions with membrane proteins. The membrane-bound protein complexes are critical for the transmission of signals across the plasma membrane. Beneath the lipid bilayer, spectrin tetramers arranged into a hexagonal network are anchored by membrane proteins. This network forms the membrane skeleton that contributes to membrane stability and membrane protein distribution. The cytoskeleton is comprised of networks of filamentous
God's
proteins
wonder.
that are responsible for the spatial organization of cytosolic components. Inside microvilli, actin filaments form tight parallel bundles, which are
This is
stabilized by
computer generated,
cross-linking
is it?
proteins.
Yes.
Oh,
While deeper in the cytosol, the actin network adopts a gel-like
But it
structure,
is based
stabilized
on
by a variety of
research
action-binding proteins.
and microscopy.
Filaments, capped at their minus ends by a protein complex, throw away from the
Electron
plasma
microscope
membrane
type work.
by the addition of actin monomers to their plus end.
It's only a few more minutes,
The
and if you want to leave, that's fine.
dynamic structure with continuous directional polymerization and disassembly.
see you in there.
I need to make a fast
Similar
announcement.
proteins induce
I'm not bailing out, I'm just gonna be out of town for the next 3 weeks.
or give rise to new filaments.
You're gonna miss a lot in
The
here.
cytoskeleton includes a network of filaments created by the lateral association of protofilaments.
You don't have any?
Formed by the polymerization of tubulin diamonds. While the plus ends of some microtubules extend toward the plasma membrane, proteins stabilize the curved conformation of protofilaments from other microtubules,
Okay, here's one.
causing their rapid plus end depolymerization.
You have an extra
Microtubules
copy of the
provide
line,
tracks
you
along
take
which
that.
membrane-bound vesicles travel to and from the plasma membrane.
You can.
The
You can have
directional
these.
movement of these cargo vesicles is due to a family of motor proteins linking vesicles microtubules.
Can you believe that?
Membrane-bound organelles like mitochondria are loosely trapped by the cytoskeleton. Mitochondria change shape continuously, and their orientation is partly
As long as
dictated
we
by
get
their
there by
interaction with microtubules.
Consecration for good.
All the microtubules originate from the centrosome, a discrete fibrous
Yeah.
structure
In
containing
biology, you learn about the centrosome of the cell.
and located near the
No,
cell.
honey, most of
Nucleus.
these people are too old to have learned that. Yes, well, that's just you and I, Gail, everybody else here. And
As
you.
small as I went.
Yeah, me too.
Containing
I was in a
mRNA
dumb kind of.
proteins into the cytosol.
That's amazing.
Look at them looking at each other.
Here, free ribosomes translate the mRNA molecules into proteins. Some of these proteins will reside in the cytosol. Others will associate with specialized cytosolic proteins and be imported into mitochondria or other organs. synthesis of cell-secreted and integral membrane proteins is initiated by free ribosomes, which then dock to protein translocators at the surface of the endoplasmic reticulum. Nascent proteins pass through an aqueous pore in the translocator.
New protein.
Cell-secreted proteins accumulate in the lumen of the endoplasmic reticulum, while integral membrane proteins become embedded in the endoplasmic reticulum membrane.
That's a highway system inside the cell.
Proteins are transported from the endoplasmic reticulum
Oh,
in
we're
the
not even looking at
It's a
the
highway
right.
system.
We're looking at
vesicles
one
that
cell?
run along the microtubules.
Yeah, it's one, inside one cell.
Inside one cell.
But see, this endoplasmic reticulum starts at the centrosome and goes all over the cell inside of like the superhighway system, and proteins move
Protein
along
glycosylation
those cells
initiated
in
in the
trucks
endoplasmic
called
reticulum is completed inside the
what is it
lumen of the Golgi apparatus.
Golgi bodies, right? And they're like balloons, and they hold the
Fully glycosylated proteins are
proteins,
transported from the
and there's
Golgi apparatus to the plasma
zip
membrane.
code destination, and so it goes to the zip code, and then the body opens up,
When a vesicle
and
fuses
the
with the plasma membrane,
proteins pop
proteins
out where they're
contained in the
supposed
vesicle's
to.
lumen are
How did it
secreted.
get the zip code?
Proteins embedded
Why
in
do
the
you know
vesicle's
about the zip
membrane
code?
diffuse
There's so
in the
much
cell
information
membrane.
here, it's
At sites of
mind-boggling.
inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor, and the consequent
And these, we're
activation
talking
of
about
the
the fingers
subunit
now on
of
the
G
outside,
protein.
right?
The activation of the G protein subunit triggers a cascade of protein activation, which in
Between
turn
the
leads to the
leukocyte
activation
and the
clustering
membrane
integrins
of the
inside
cell wall,
lipid
of
reticulum.
the,
A dramatic
vessel.
conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation.
remember the
Visual
chemokines that
signaling
started
events
this whole
cause a
cascade?
reorganization of the
Because
cytosol,
they were,
resulting in the spreading of one edge of the leukocyte.
then,
The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte extravasation.
pretty interesting. Just simple rules.
I'm sitting here thinking, wonder what my vitamins do all that.
Just simple rules.
Well, I can
It's
tell
not
you-
just simple rules.
I
It's
can tell
an
you.
incredibly vast and incomprehensibly complicated design.
Oh, yes.
Thank you very much.
Thank you. That was quite eye-popping.